Okay, welcome back to the Classics of the Immunology Journal Club. Before we get started, I just want to remind you to like the video on the YouTube channel and subscribe. Click on the bell to get notified for more. Check out my website, kendallasmith.com. On the writing tab, you'll find all the classic papers. You can go back to 1798 with Edward Jenner and then work your way forward. Today, we are in 2016. Not bad, huh? We're almost up to the present time. And the paper that we're going to review is by Katrin Klocke, Shimon Sakaguchi, Richard Holmdahl, and Kaya Wing. This is from the Karolinska Institute. It was published in PNAS in 2016. And the title of this paper is The Induction of Autoimmune Disease by Deletion of CTLA-4 in Mice in Adulthood. So the background on this paper, not to recapitulate everything in this whole regulatory T cell area, the standard CTLA-4 knockout that is knocked out during embryogenesis is lethal. The mice are born, they're fertile, the mice are born, but they really go downhill rapidly and they die within 3 weeks. And that was published in 1995 on the CTLA-4 knockout mice by two different groups, actually. Now, last week we did one of Kaya Wing's papers from 2008. So this is 8 years subsequently, where she did a Treg specific CTLA-4 knockout. And found that the knockout of this particular gene was still lethal, but it took the mice 10 weeks to all die. So there was a delay in terms of that whole thing. And as you recall, CTLA-4 is expressed constitutively on regulatory T cells, together with FOXP3 in the nucleus, CTLA-4 on the cell surface. But it's also induced by T cell receptor activation on antigen activated T cells. And it's been shown that interleukin 2 is the stimulant at the genetic level for both CTLA-4 and FOXP3 expression. So both of those genes are parts of what I call a negative feedback loops that regulate the immune response upon antigen stimulation and activation. In between 2008 and 2016, there was a paper that I want to draw your attention to that's on the website. It's one of my papers, together with, it's really from the laboratory of Gregoire Altan-Bonnet from the Sloan Kettering Institute in New York, where we looked at and quantified the expression of the IL-2 receptor, in particular the alpha chain, CD25, and found it to be markedly upregulated on regulatory T cells. And as a consequence of that upregulation of the alpha chain, the affinity of the trimeric receptor, alpha, beta, gamma, was increased up to 150-fold, which gave the Treg cells a huge advantage on what Gregoire voiced as the tug of war for IL-2 between Tregs and then the conventional antigen activated T cells. So the Tregs were always going to win, particularly when the concentration of interleukin 2 in the milieu surrounding the cells was on the low side, because the KD, or the equilibrium dissociation constant, was in the range of the femtomolar range, 10 to the minus 15th molar. Pretty good affinity. Not bad, as the French would say. In 2015, just a year before this paper came out that we're going to review today, Arlene Sharp from Boston and Harvard published a paper, a very similar paper to this one, and found that when she deleted CTLA-4 in adult mice, it did not precipitate systemic autoimmunity. There was generalized activation of the T cell compartment within the immune system, but without significant autoimmunity, according to her. So this paper is in opposition to that. And in the introduction of the paper, Kaya Wing points out early on in the introduction that anti-CTLA-4 treatment, blocking anti-CTLA-4 in adults with cancer, humans with cancer, often results in significant anti-tumor immunity and disappearance of the tumor. But at the same time, there's also significant increase in the incidence and severity of autoimmune phenomenon that occurred, and to the point where that became the major adverse events that the oncologists were confronted with, which they really never had been confronted before because they didn't have to deal with the immune system, not the way they do today, that's for sure. So the question is, why didn't Arlene Sharp's paper find anything like that when she knocked it out in an induced knockout in the mouse? And Kaya Wing goes on to point out in the introduction that CTLA-4 has has been shown over the years to have two different effects, inhibitory effects on the immune system. One that was autonomous or intrinsic, and due to the fact that CTLA-4 would decrease T cell receptor signaling by inhibiting the activation of cytokine genes, particularly interleukin-2. In addition, there was a non-autonomous or an extrinsic mechanism that CTLA-4 had been found to have, and that was it had a much higher affinity for the ligands CD80 and CD86 than did CD28, the co-stimulatory molecule on T cells. So it dampened down co-stimulatory signaling because it competed for the ligand CD86, and it was an inhibitory kind of a situation. So that's given as the background, and so therefore Kaya wanted she knew that the congenital knockout from 95 of CTLA-4 was rapidly lethal, making it, which was interesting. So the mechanism of that, but it made it very difficult to study because the mice died before you could really, mount your attack on this whole problem, and therefore she decided that in order to really study this phenomenon, she needed to knock out this gene in adulthood. The 8 years between the time that she was with Sakaguchi as a postdoc in Japan and 2016, she came back to Sweden and started her own lab at the Karolinska Institute in Stockholm. And this is one of the first papers to come out of her own lab. So the materials and methods in this paper are important because, because of the fact that the way CTLA-4 became constitutively expressed on regulatory T cells, that occurred during development, after birth, and you really couldn't specifically knock out CTLA-4 only in T regulatory cells from an adult. So basically, she did the next best thing, which was to knock it out of both T regulatory cells as well as conventional T cells if and when they became antigen activated. And in order to do that, the sequence of events was in terms of constructing of the mice in order to be able to do an inducible knockout. First of all, the strain of mice that had CTLA-4 floxed and another strain of mice that had the Cre enzyme with an estrogen receptor promoter on it. They were backcrossed onto a particular strain of a mouse, C57 black 10Q. And they did that for more than 6 generations. And then they crossed the Cree ERT2 mice with the CTLA-4 floxed mice, creating an induced knockout. Because then, if you could wait till these mice grew up and they were fine and so forth, and then you could induce the deletion of the CTLA-4 gene by treatment of the mice with tamoxifen, which would bind to the estrogen receptor that would activate the Cree recombinase, and that would then cut out the CTLA-4 gene. Now, the control for that was just the wild type bred onto the CTLA-4 flox background. And then to induce the deletion of CTLA-4, what you do is you treat those mice intraperitoneally with tamoxifen suspended in corn oil because it was hydrophobic. And you treat the mice, you inject them at day 0, day 1, and day 5. And by day 6, as we'll see, the CTLA-4 gene was completely knocked out. Now, she also generated congenital CTLA-4 knockouts by crossing a CD4 Cree under a CD4 promoter and CTLA-4, the floxed CTLA-4. So that's the essence. Then there were a lot of immunological methods that we've talked about for many of these papers. So we don't need to go back through those again. So let's go to screen sharing. And in figure 1, this really shows the efficiency of the CTLA-4 gene deletion in the adult mice, and the fact that it generates a lymphoproliferative situation and an autoimmunity similar to the congenital knockout mice, different kinetics. So in A, what we have here on the left-hand side is the wild-type control, and we're looking at CTLA-4 versus FOXP3, and here's the induced knockout before treatment with tamoxifen. And you can see that almost about 40–50% of the FOXP3 positive cells are CTLA-4 positive. So then this is on day 6 after the day 0, 1, and 5 treatment with tamoxifen. Wild type control is unchanged, whereas the induced knockout, IKO, essentially the CTLA-4 double positives with FOXP3 are absent. And of course, the FOXP3 negative cells, which are the majority of the cells in the mouse at this stage, T cells, are CTLA-4 negative, because it's only when they become induced by the T cell receptor that they become positive. Now, as a consequence of this knockout phenomena of CTLA-4, these are mice that have been allowed to grow up past puberty. And then after the CTLA-4 depletion, if they look at the survival of the mice, the wild type, 100% survival up to 100 days, and the induced knockout, also survival up to 100 days. In contrast, if you look at the congenital knockout, as we've seen before, here's the congenital knockout. Essentially, they're dead by 4 weeks. The wild type is fine up to 60 days in this particular figure, whereas 16 of the congenital knockouts, these are the data here. And the other thing about this is that in C and F, what you see is massive lymphadenopathy and splenomegaly here. And so here, the open singles are the knockout, and the closed are the wild type. And you can see the spleen it's elevated, and it's markedly elevated in the lymph nodes. And this is the induced knockout. And here we are, just let me see, 1 day, they are just at 20 to 20 days before they start to really go off here. And you can see that there's a marked increase in splenomegaly as well as lymphadenopathy in the congenital knockout mice. So then they also found an increase in the immunoglobulins in these mice. Now, if you go on to figure 2, you knock out CTLA-4 even in the adult, and you start to very early on afterwards, you start to see immunopathology essentially. So in figure 2, what we see is the spontaneous development of autoimmunity in the induced knockout mice. And so in A, up here, these are histological specimens of different tissues and so forth. In A, we've got the wild type on the top row. Then we have the induced knockout. And the induced knockout is 16 days after the depletion of, well, no, that's in the congenital knockout, 16 days, just before they start to die. And the induced knockout is at 8 weeks after the depletion. And here's the congenital knockout. And so what you can see is that in the lung, the wild type is fine, whereas the induced knockout is starting to have peribronchiolar infiltration of inflammatory cells. And of course, the congenital knockout is much worse off than the induced knockout. Salivary gland, it's the same story. We have foci of what look like lymphoid follicles are developing in the salivary gland, and here's a diffuse infiltration of the round cells. Stomach is the same situation. Pancreas as well, with severe disease in the congenital knockout, whereas liver and heart were fine in the induced knockout, but the congenital knockout, there was a severe pathology. If you then went on and if you look at this is schematic of the data of involvement by inflammatory cells. Wild type is negative. This is the induced knockout. This is the lung, salivary gland, stomach, pancreas, heart, and liver. And there's a varying degree of involvement histologically at the time intervals that they studied them. But the congenital knockout was more severe than the induced knockout. If you then look at the kinds of cells that are involved in these infiltrations, this is staining for anti-CD3, and this is for FOXP3. This is the lung, salivary gland, stomach, and pancreas. And you can see that it's in the dark brown or the brown staining of T cells involved in the lung, diffusely in the salivary gland, the stomach, and the pancreas, and regulatory T cells, FOXP3 positive cells, also proliferate in this syndrome and infiltrate the organs. So then if we go on to figure 3, I just want to focus down on the D portion of this figure, because what we've got here, this is looking at induced knockouts 1 week after the full depletion of CTLA-4 in these induced knockout mice. And it's doing intracellular staining after stimulation of splenocytes with PMA and ionomycin, looking at interferon gamma, interleukin 16, IL-2, IL-4, and IL-10. And with the exception of interferon gamma, you can see that these cytokines are all turned on in these cells. So these cells are not only are they proliferating and you get increased numbers of them, but you also have they're activated, and they're activated by virtue of the fact that now they're expressing their cytokine genes. So that's the essence of this paper. It's an extensive paper, And in PNAS, you're limited to like 3 or 4 pages, but she's got multiple subfigures in her figures and multiple experiments and so forth. And if you're interested, you should really read this paper carefully. But I will stop the screen sharing at this stage. Okay, so let's go on to the discussion at this stage. Kaya Wing was very concerned that Arlene Sharp had her paper out a year before this paper came out, this one on PNAS. And Arlene's paper was in the JEM, and that's also on the website, so you can take a look at that and read through there. What Kaya points out that the discrepancy between her findings and Arlene Sharp's findings could have been due to several different situations. One is that the genetic background of these mice was different. These are all inbred strains and so forth. The Cree recombinase was in the 2 different experiments were under different promoters. And how efficiently that promoter turned on Cree recombinase could very well have resulted in the efficiency or lack thereof of the depletion of CTLA-4. And to my way of thinking, that's probably the most logical kind of reason that they could be different in terms of their findings. Also, they were different histocompatibility type. One was Q, the other one was B. But the other thing that Kaya Wing points out is that they both had severe lymphoproliferation and increased regulatory T cells that were CTLA-4 negative in both of these studies. The one was just the difference. The major difference was the lack of autoimmune phenomena. And of course, that was also in Arlene Sharp's paper, and that was counter to the human findings in patients that were treated with anti-CTLA-4 because they suffered from adverse events and severe autoimmune phenomena. So the other thing that Kaya points out in this paper is that whether this CTLA-4 depletion in adult mice and the effects of those, whether it was primarily on the conventional T cells or whether it was on the FOXP3 regulatory T cells, she couldn't really speak to that in this particular paper because they were knocked out in both of them. The other thing that is evident here is that CTLA-4 deficient regulatory T cells could probably also use alternative pathways to depress immune responses, one being the depletion of IL-2 through binding to the IL-2 receptor, internalization and degradation of IL-2. And so I think it's still unsolved as to all of the molecular mechanisms that could be at play that allow regulatory T cells to act as basically a negative governorship on T cell activation and proliferation and the discrimination of autoimmunity, self, non-self discrimination, and autoimmune phenomenon, as well as anti-tumor phenomenon. And so the jury's still out, and we still have more. We still have more to do. And this is job security. So that's the end of that. And don't forget to like the video and subscribe, click on the bell, and come back for more. Check out the website. Read all the papers. Very important. So thanks for listening, and see you next time. Thanks. Bye-bye. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club. You can't go wrong.